Hello, everybody. Good morning to everybody. We are going to start the second day of this Congress, and we will start with Professor Dame Janet Thornton. Let me introduce her. It's a great honor for me to present you. Professor Dame Janet Thornton is a senior scientist and director emeritus at the European Bioinformatics Institute and formerly director from 2000 to 2015. ABI, that institute, as Janet said in a conference, provides biomolecular data for everybody around the world. It's really very important. So she's one of the world's leading researchers in structural bioinformatics, using computational methods to understand protein structure, function, and evolution. She played a key role in Elixir, sorry for my pronunciation, that is the pan-European infrastructure for biological data since its inception. That is also a really important institute and and she's the key role in that infrastructure. Professor Thornton is a fellow of the Royal Society, a fellow of the Academy of Medical Science, also a member of the European Molecular Biology Organization, and is a foreign associate of the US National Academy of Sciences. She has contributed to the science with more than 500 scientific papers, but all of these are important distinctions and numbers, but they don't really explain her contribution to science, to the understanding of protein, both in normal conditions and in diseases. The tools and databases he has developed are used worldwide for basic research, academia, and pharmaceutical company. I have an anecdote as well. I have read your paper about CAP database in 1999, and I use it since then. For those who do know, this database is a database of structural classification of domain of proteins, and is really a tool of an enormous use nowadays, 20 years later of her development. So there is no way to summarize a life dedicated to science, particularly to bioinformatics, in a few words or in one page. So I'm very grateful that you give us the opportunity to listen to you, and above all, that you serve as an example for so many young ladies scientists who will be listening you just in that moment. So thank you very much again, Janet, for being here. It's a great pleasure and honor to have you here. And let's start with your talk that is about computational enzymology, the structure, function, and evolution of enzymes. Thank you very much again.
Thank you, Christina. And it's, for me, a real pleasure to be here. I think you said I was a good example. I think, Christina, you are a wonderful example of a bioinformatician and a female. Yeah. And I love this idea of having a conference where all the speakers are women. This is fantastic. Yeah, I think it's very unusual. It's such a shame not to be there and to enjoy that aspect of it as well as the research side and then presenting the work. So unfortunately, my internet in the place I am is not as stable as I would like. So I'm going to turn off my camera in the hope that,
everything will be
okay. And now, shall I share my screen?
Yes, please.
Good. Can you hear me?
Yes. Yeah. We can hear you, and you maybe want to put it in the presentation mode. We can see your slide.
Okay, brilliant. Okay, all
perfect.
Good?
Everything perfect. Okay.
Right, fingers crossed that we stay in touch. So I'm going to talk to you about computational enzymology. And as you know, enzymes are really important molecules. They were first used, the first evidence that they were used in food production occurred many, many years ago. So in 7000 BC, it's known that yeast and fungi were used in food production. And you can see some of the things here, bread making in the ancient world, and also sake making, alcohol, in Japan. But the
Oops, I think we are having a problem with Janet internet.
actual active ingredient as 1897 that cell-free fermentation was possible in not just, well, in cells, but in the fermentation that allowed you to use these properties for fermentation. And during the 1900s, the curly arrow diagrams were generated, and hello, can you hear me?
Yes, we can.
Yes, we can.
Sorry, I'm paranoid. We saw the diagrams of the whole metabolic pathway, which was a phenomenal piece of work. But also, we began to understand much more about the proteins, the actual enzymes that perform the catalysis. And here you see the structure of hen egg white lysozyme, one of the very first enzymes. And of course, since then, now we know about thousands of enzymes in this century. About between 25 and 50% of proteins encoded by genomes are enzymes. We have sequences for 32 million enzymes. We have structures, and this is now a few weeks old, so this will be out of date already, for 45,000 structures. And we know that these enzymes perform many different sorts of reactions. And during that time, quantum chemistry methods have been developed to analyze enzyme mechanisms, and also a few successful enzymes have been designed. But despite all this knowledge, there are still questions about how exactly some enzymes perform their mechanisms, what the green chemistry is, how does enzyme function evolve? Can we predict the transformations performed by enzymes and the mechanisms by which they do those transformations? And can we predict specificity in function, the sequence and structure? And the answer for many of these questions is still in part, but certainly not completely. And of course, understanding enzymes, whilst it's academically a very interesting area, actually it has many applications in drug design, in enzyme design, obviously, in pathway redesign, and in making new enzymes for the green chemistry that the world really needs. And wouldn't it be wonderful if we could have plastic processing enzymes and clean up the oceans? So these areas are very, very active at the moment. And I think we will see another boost of activity in enzyme research. We also know that enzymes at the moment constitute about 54% of all drug targets. And of those, 75% are human enzymes that are targeted, as one would expect. And we can see these targets go across all the different sorts of enzymes, the sort of 7 different enzyme classes that we're familiar with. And we can see Of course, the majority are in the hydrolases, the oxidoreductases, and the transferases. And of course, there are many drug targets, enzyme targets, which aren't enzymes, but there are many that are. And so again, understanding these enzymes better will allow us more, I think, to develop better medicines. And in fact, if we look at the new drug targets that were identified in 2020, 5 out of 13 of those are enzymes, where we know the mechanism of action. So this really means that enzymes remain very important biological molecules. So what's our goal? To understand how enzymes work and how they evolve. Can I just check that you're still there?
We are.
Yes, we are. Yes, we are, and we are listening to you very well.
Okay, I'm just worried. It's like speaking into the ether and I can't hear or see anything.
We just close our microphones to make you feel comfortable.
Absolutely. So in our computational enzymology, our goal is really to understand how enzymes work and how they evolve. And over the years, Christina mentioned some of our cath data, and we worked on many different aspects of enzymology. We've looked at evolution and how the families change. We've looked at specific families. We've looked at catalytic residues, transformations, ligand binding, pseudoenzymes, always really relating what we know to the structure, the three-dimensional structure of the protein. And in particular, in doing this work, we had to gather the data out of the databases, and we created something called the MCSA, the Mechanism and Catalytic Site Atlas database, where we gathered the information that we needed from the three-dimensional structure. So I'll talk about understanding catalysis. I'll talk a little bit about characterizing flexibility and plasticity of active sites, and a little bit about trying to predict enzyme mechanisms. So to start off with saying a little bit more about the Mechanism and Catalytic Site Atlas. And the people who've worked on this, really, this started off with Gemma Holiday, who was taken up by Antonio Ribeiro, who is currently responsible for it. And the database really, first of all, information about the reactants and the products. This sort of knowing what the substrate is and what the products are, that's critical, and it's this reaction that, of course, defines the enzyme commission number that we're familiar with, the EC number, where each enzyme is defined according to it's a hierarchical classification with 4 numbers, where the first number defines, if you like, the overall class of enzyme, and the last number defines something about the substrate usually. But it also importantly includes the information about the protein, the enzyme sequence and its relatives, with connections to the PDB files and the cath files and the species, of course. It tells us about cofactors and what cofactors are used. And it also tells us about which are the catalytic residues. And this was where we started, because of course, without protein structure, you really cannot define the mechanism and the catalytic residues effectively without that information. So this is all freely available. In fact, we've just done an update of the MCSA, and we now have over 1,000 entries of different enzymes in this database. So with the database, we can begin to ask questions. And one of the questions we wanted to ask was, well, how can we link the amino acid residues to the chemistry that they perform? And so we have, this is now the old data, as I said, now we have more information on the mechanisms. But here we had information on about 3,500 residues and mechanism steps, and also we assigned to each residue a catalytic role. So here we have the 20 amino acids, and the data.
Janet, we are losing you for a few seconds.
Hello?
Yeah, now.
Hello.
Yeah, maybe this slide could be again, because we lost you for a few seconds.
Okay, I'm back again, Emma.
Now it's okay.
Okay. Amino acids here. The histidine, which is the most common amino acid involved in catalysis, as well as asp, glu, glycine, arg, and cis. Those are the sort of classic amino acids. And if we normalize that, if you like, by the frequency in the protein sequence, we can see that, of course, the histidine, aspartic acid, and glutamic acid are the most common, and cysteine is also very common, given, of course, that it's much rarer in an amino acid sequence than the other three. We can also look at the main chain. So, because very often the main chain, the NH and the CO group of the main chain participate in catalysis. And when we look at those, which amino acids are involved there, we see that the most common are glycine, serine, and alanine. And of course, these are the smallest side chains. Glycine doesn't have it, well, apart from the hydrogen, doesn't have a side chain. And glycine absolutely predominates in terms of its importance here for catalysis. And that really is a case of accessibility, that the main chain is much more accessible in these relatively small amino acids, and cysteine is also a relatively small side chain. And so we can look at these catalytic residues that are reactants. And here we can see we have the different sorts of reactants that we can have during catalysis. So there's proton acceptor, I hope you can read this, they're rather small, and proton donor. And the first thing that struck us when we looked at this is the absolute dominance of proton acceptors and donors. And effectively, sometimes I think that the whole of proteins, the enzymes, they just work by shuttling around protons at some level, apart from, of course, there's the electron shuttles as well. We can classify some as nucleophiles and nucleofuges, and the proton relay, which is sort of similar to this, and then the electron acceptor and donor, and single electron and the relay. So you can see that actually, in terms of the reactant, the actual mechanism that these amino acids use, then you are really dominated by certain types of reactants. And these are the, here we can see the reactants, and we can see our 20 amino acids along the top. And the number of times that a given amino acid performs a given role. So what you can see here is that HISP, GLU, and ASP in particular are used as proton acceptors and proton donors. And cysteine, whilst it is also a proton acceptor and donor, is much more important in the sense that it is a nucleophile and a nucleofuge. So from that point of view, and the lysine and the serine, which again can be proton acceptors and donors, but they also are frequently used as nucleophiles and nucleofuges. You can see as well that if we're looking at electron pair manipulation, acceptors and donors, then it's lysine that dominates in this respect. And when we look at these more uncommon reaction roles, then we see that the cysteine, if you like, comes into its own and becomes tyrosine and even the main chain glycine occasionally. So when we then can say, okay, let's just look at one of these roles, for example, the nucleophile. Here you can see the nucleophile level, and you can see that here these are the frequency of the residues that are able to have a nucleophilic role. And what actually continues to amaze me a little bit, have quite a limited scope in terms of what they can do catalytically. Of course, we haven't, I don't talk about the cofactors here, but from the side chain, most of the amino acid with lone pairs can be nucleophiles, but some of the role more commonly than others. And so another aspect that we can look at is to look at enzyme active sites in 3 dimensions. And one of the questions is to what extent they are flexible and to what extent they are plastic during evolution. So they change during evolution. And so Yannis Riziotis has created a pipeline that's called CSA3D that reports on the active sites of enzymes. This isn't published, it's not available yet, but it will be attached to the MCSA. And what he's tried to do is to really characterize how flexible, how plastic enzymes active sites actually are. And using the MCSA, we've got active sites for about 900 different enzymes. And so we look at the flexibility within a single protein, but also the plasticity between proteins. And we do this, we compare structures for which we need the sequence alignment, the structural alignment, and identification and classification of the ligands. And so one of the questions we've been trying to ask is, we know that as the sequence identity between 2 proteins decreases, then the structural similarity decreases in line with that. Our question really was, if you look at the sequence identity, how will the active site similarity compare? So what are the factors that control the active site? And what we find is that the overall structural similarity decreases beautifully as the sequence similarity, the overall sequence similarity, sorry, I think this is the wrong way around. The overall sequence similarity increases, that's right. As the overall sequence similarity increases, then the root mean square deviation between any 2 structures increases. If we look at the active site only, so the catalytic residues, what we find, of course, is that this correlation is much, much less strong. And you can see this is just at 2.5, and as the overall sequence similarity, even when you have very low sequence similarity, the active sites are still very similar. They're very conserved, which is, of course, what we already know. But we also know that many of these active sites in the protein data bank will have identical ligands bound or different ligands bound. And when we look at the identical ligands, we see that they have very low root mean square deviations. This is just for the catalytic residues only. And so it's less than 0.5. And if they have different ligand bound, of course, that standard deviation increases, but it's less, it still remains very low before the catalytic residues. The active sites are really the rocks in the core of the protein. And so if we look at a particular example, we can see here, this is the camphor 5 monooxygenase. It's part of the cytochrome P450 family, which is a very large and diverse family. And you can see there are many different enzyme functions within this family. And this is the reaction that it does. And you can see this particular flavor of P450 is found predominantly in the bacterial families. And when we look at it, what we find when we compare, so this then shows a plot of the active site, we can see the heme cofactor here, and the colored residues that you can see, the 6 colored residues have been identified as catalytic residues. And what you can see is that most of those catalytic residues actually are extremely well conserved in their geometries. The only exception is this arginine, which is on the surface, that basically acts as part of the proton relay and has different conformations. But the catalytic residues themselves are very, very well conserved. And so similarly, when we then go and look at the gly-lu-cis, the other thing I should say is that we can think of these as being two modules. Here we have one module, and here we have another. This is involved in the heme interactions, and this is involved in the proton relay to take the proton out of here, the gly-lu and the cysteine are conserved, not so much the lu, but the cysteine certainly, whereas the proton particular, which we saw was flexible, this is much less conserved over evolutionary time. And we can look at the distribution of the catalytic site residues across here. And I don't know whether this is going to work. Can you see that going round?
Yes,
Yes.
nicely.
Yeah. So you can see the conservation of these residues. The 5 residues are extremely well conserved in structure, whereas this arginine, and you can see there are some strange different residues. This is partly, ignore that because there are some problems here, but it basically emphasizes the flexibility. And when you look at all the active sites over all these protons, all these proteins in this family, you get a whole a variation of root mean square deviation. And you can see when we're looking at them here, this just shows for the different residues in this family, how extremely well that, of course, the arginine here is the one that doesn't have any ligand contacts, and that is important because this is the ligand here, the camphor ligand here, and that lack of contact to the ligand means that the flexibility or the variation in the structure doesn't. And also see that the has different levels of accessibility compared with what you might expect. Constant their relative solvent accessibility, whereas the arginine varies enormously between that. So what we've got, and this is really a presentation of something that's not yet completed, because we've been looking across many different enzyme families and generating root mean square deviation by comparing all the examples that we've got. And this is just for enzyme families where we have quite a lot of entries in the PDB. And you can see when you look at these root mean square distributions, some of them, a lot of them, are very well conserved, that you can see here. But others show perhaps two states where you have some in one state, some of the examples, and another in another state. That sometimes corresponds to whether there's a cofactor bound or not bound. So interpreting these root mean square distributions actually is complicated, and we need to look not just at the RMSD values, but also about which ligands are bound and which ones are ligand free. So it's very much work in progress. So the other area where we are working is looking at enzyme mechanisms. And of course, the enzyme mechanisms have long been represented as these curly arrow diagrams here. So this just shows one example. And within the database, within the MCSA database, Antonio Ribeiro, we have of different enzyme mechanisms. And with the recent update, in fact, we have a very high coverage of those mechanisms, which have been determined and validated to some extent, but higher now than that 80%. It was over now at 90%. And we can see that actually about 96% of catalytic residues are polar, 70% are charged, and 70% of the reactant roles are these proton acceptors, really quite limited. The cofactors bring some extra oomph to the problem. But if we look at the side chains, it really is quite. Given what we know, maybe now was the time to use an informatics approach to suggest or predict likely mechanisms for a given structure. So this is what we've been working on. We know the enzyme reaction, and we know the enzyme mechanism. So in this case, this is glutamate racemase, and we can see the steps involved. First of all, the aspartic acid grabs this proton here, which goes onto the asp. Then the serine is primed. It can then attack the NH3 here of the substrate. And then that means that you change the electronic state around here, and that's what allows the cleavage of the bond. And then this cysteine on the other side accepts the proton from from the substrate. So and then it goes back to normal. So the question is, can we, given if we know the enzyme site, we know the reaction, so can we create an algorithm that predicts possible enzyme mechanisms based on our knowledge of how proteins generally perform catalysis? So this is the question that Antonio has been working on now for about 18 months. So what he's doing is actually extracting these rules that capture how this mechanism occurs. So this is a sort of one way of looking at a rule, where you have the SH group, CSH, with the CO minus, and one rule is that becomes a COH and a CS minus. So we have stable reactions that we've already observed. So what we do is we look at every mechanism we have in the database. We only use those that we've given a 3-star rating. That means we're confident they're correct. And from these, we have about 2,700 steps where we have this information collected with 17,000 odd curly arrows. And from that, we can extract about 7,000 rules, and we add the additional constraint that these rules must be stable. So they result in molecule, if you like, molecules which are relatively stable. And so this is captured in the SMARTS representation, which is a cheminformatics representation of active sites. And we capture information at the 1D level, which is the reaction, at the 2D level, where we include all the protein, etc. And we can plot it out in space, but also we use the 3-dimensional data. And so what we do is we take an enzyme active site and we iterate over all the rules and generate multiple configurations of that enzyme active site. And you can see those we can represent as a network, actually. So we have the reactant at the beginning, the 2 possible configurations, and then one of those goes on to produce the other one and eventually gets to the product. So you could iterate over these rules many times to try to predict how this actually works. And here we can see the network that's generated for beta-lactamase. And in fact, the actual mechanism that we observe is shown by in this space. So we're really working on trying to improve this prediction and to learn how to do that. Created the algorithm. We know how to look at it and to see whether it's right or wrong using this network representation. And Antonio is in the process of working through. I should say that when we actually have this, we can automatically generate, you can see here, this is the glutamate racemase where this reaction occurs. So you can generate a video of these mechanisms. And I kind of feel that that's quite important for us to visualize what might be going on. So, of course, one of the things that's happened recently that I could talk about much more is the fact that now we have models for most of the model organisms from AlphaFold. So the AlphaFold database was released from the EBI now several weeks ago. We have predicted structures for 365,000 proteins, and we have almost complete coverage for 21 model organisms. This gives very good coverage But importantly, it also tells you where the confident predictions are and where the not so confident predictions are. So this reliability measure is really important. And with this sort of prediction data, then we can use our tools for enzyme mechanisms and test whether actually these new models are enzymes and also how they might work. We'll see whether that comes about. So our future work then is mechanism prediction, the analysis of the active site configuration. We're constructing temporary libraries of templates, looking at flexibility and dynamics. And obviously our hope is that this will contribute to our ability to repurpose or redesign from scratch new enzymes and understand how a given family evolves to perform multiple functions. So if I could just acknowledge the people, I've talked about Antonio's work in the MCSA, I've talked about Yanis's flexibility work, I've not mentioned the work of Roman, John and Neera, but also I think I should acknowledge Gemma Holliday and Nick Furnham, who were very much involved in the MCSA and its development. And of course, the work on the protein evolution is all with Christina Rengo at UCL. And perhaps I could acknowledge EMBL and the Wellcome Trust for their funding. So I hope the internet hasn't caused too many interruptions in this and that you can at least get a feel for what we're trying to do. It's still very much work ongoing. So thank you very much for listening.
Thank you very much, Janet, for such an amazing talk and enlightening and inspiring and very educational. I think it was a really amazing talk. Maybe we can see you. Do you think
we cut out? Sorry.
If you feel...
It's all right. Carry on.
I think we've lost you for a few seconds.
Okay.
Now we are hearing you.
Can you hear? Yes.
Yes.
Shall I? I'll stop sharing. Shall I? Yeah. Okay. So I've stopped sharing.
Okay.
Yeah, we hear you. I don't know if you can open your camera or is
yes,
the internet is... Okay, now we can see you. Thank you.
Much better when you can see people.
Yeah. Janet, it was such a nice talk. Everybody's telling the same in the chat and in YouTube and the Q&A's tab. So I was wondering, we have many questions, but I have the privilege to be the chair, so I will ask you one question first. I was wondering, just you talk about at the end of the talk about this AlphaFold 2 that we are all knowing those days. And I had the idea that it won't be able to capture catalytic site flexibility and plasticity. But
so it's very interesting actually, because AlphaFold generate you can. There are I don't know if you know, there are servers now that you can use.
Yes.
The database only has one structure.
One model.
But normally they generate 5. And sometimes those models have different conformations. Sometimes they're all very similar, but sometimes they are quite distinct. And so it might be that they capture some of the alternate conformations. I honestly don't think that has been studied in enough depth yet, but I do know that some people who are using the models for EM interpretation find that it's useful to use the different models, because if, you know, in an EM, you often have different conformations, and sometimes they find that different models fit the different images
Yes.
better. So I don't know. I mean, AlphaFold is very weird. It hasn't solved the protein prediction problem at all, the protein folding problem. It tells us
Yes.
nothing about how proteins fold, and yet it captures these the predictions are, for some proteins, remarkable.
Yes.
Many of those obviously use homologs, and I think that's important. But even for those, they've actually managed to improve the accuracy of the what I would call homology built model. So I think the impact of this is going to be very interesting going forward. It's in fact, most of the structures which are drug targets, most drug targets we already have structures for. So in fact, AlphaFold doesn't add very much.
Yes.
But maybe for some weird and wonderful model organisms that cause diseases, that will be helpful. And I think it just changes the way that we think. If we think, oh, well, we can try and predict a structure,
yeah,
and it could be very accurate. I think it's changed the way that people think. I don't know what you think.
no, I think that even if they can provide us with the fold and the structure, I thought that this fine-tuned position in the active site maybe is not captured by
well, but in a funny way, because you know, which is why we started looking at the catalytic site residues, because they are remarkably well conserved.
Yeah, that's true. That's what you've shown.
Yeah,
And it was surprising me.
They are like rock, and it's because of the entropy. Because for catalysis, it's got to be fast, and so you don't want these molecular movements because they are slow. And so, you know, some of it, I know that, you know, the substrate has to get in and get out, and there are changes, but very often they're not in the catalytic residues. They're in these ones that are stuck on the outside, like the one I showed you.
Yes, it's arginine that moved more.
Yeah, yeah.
So I would like to speak with you the whole day, but I have some questions from other people to tell you. Yeah. Priscilla Esteves asked, thank you very much, Janet, for that excellent talk. I love it. And she's asking about non-catalytic residues rule for stereospecific enzymes activity. In your opinion, what are the perspectives on this field?
I'm not sure I understand the question. Can you repeat it, Anna?
Yes. She says that what about non-catalytic residues rule for some stereospecific enzymes activity?
Non-catalytic role
for stereospecific enzyme activities.
So does she mean, do you think, I don't know, you probably don't know either, the binding site residues are often not the catalytic residues, but the binding site determine what binds, and that gives often the stereospecific. So it's not so much in the actual mechanism, it's more in the how it fits in. And so catalysis to me is all about the residues being in the right place at the right time. It's like a dance. I always think of it like a dance. I don't know, in the right place at the right time to make it work. I think the internet has frozen a little.
Yeah, we are having problems hearing to you. But I think we understand the idea.
Yeah, maybe we can try with the camera off.
Internet is not in our side today.
Okay, I think
Now, yeah, now we can
Now we can.
see you a little bit for a while.
yeah.
So, but I think we understood your explanation. I'll go further with another question. If Mercedes Didier asks, I have a question. I work with promiscuous proteins, and I notice when trying to link information that annotation many times is missing. And also classifying protein as promiscuous by easy number was impossible because of that. So, do you think considering promiscuous activity can change the knowledge about amino acid rules or the predictions we have made? Have you considered promiscuous cases?
Yeah,
Um,
actually, no, that actually is my question, Janet, because I have used many times catalytic site atlas, and trying to link the knowledge we have in with the structure to the function based on EC number is so complicated. The data is there; it's missing, and so.
No, absolutely. I think the promiscuity. So there are two. I think obviously promiscuity is really important. I mean, I think to me this is probably the big unsolved problem, as well as predicting mechanisms, but also given a protein, what's its substrate, and does it have multiple substrates or not? And in fact, I don't have an answer to that. We don't. So have you been trying to link things through the Brenda database? Is that what you've been doing?
Yeah, in my PhD, I was working with promiscuous protein, and actually I was trying to link the information about evolutionary aspects and three-dimensional aspects, and trying to my best way to get to something was with the evolutionary aspects, because we don't have enough information about the catalysis, sometimes about the promiscuous catalysis. So the role of the amino acids,
as well, I think, is if you have
missing.
a family of enzymes, and I didn't talk about this at all.
Sorry. No, now we hear you.
Yes, I think she said that you didn't study that at all.
I only get one or two words. But promiscuity is very common. I think we need quantitative measures for promiscuity, and I've been trying to persuade people in my lab to work on this, and I've failed, I have to say, but I think it's really important.
We can collaborate, because actually I will be very happy to upgrade the catalytic site atlas with promiscuity aspect, because I have built a database about promiscuity. And you know that I'm merging all the information because it's that important. I think that we can try to find the way to link this information by annotation and make it this annotation.
I mean, it is quite, when we started looking at this flexibility, there are so many, because the conformations depend on which ligands are bound, and you bind a different ligand, and then not changes so much in the catalytic residue. I mean, in fact, they're all pretty well conserved, but I mean, changes in the binding site are just critical. And so we have to extend it out. It's not good enough just to look at one thing. We have to try and, but there is the problem. If you say this enzyme is not, this only catalyzes one molecule. Of course, have you tested all the other possible molecules?
No, actually, my conclusion from my PhD is that all proteins are putative promiscuous proteins, because we haven't tested them all. Oh, we lost her. Sorry, Chris.
No.
I think that's probably into what the binding site looks like.
So I think there are many questions.
Thank you.
Lot of people interested in promiscuous enzymes. Maybe you can make a chance to study that. This and enlightening us with that as well. So I think we have run out of time, and we want to thank you again many times, as you have given us a great talk.
Janet, are you still there?
I think it's a very bad connection today.
Yes, I'm here.
Okay. We thought we lost you.
So it was difficult, but by internet. But don't worry, it was a perfect talk and very nice talk. And as I tell you, educational and enlightening and inspiring talk.
Wow, thank you very much. I'm really sorry about this.
No, don't worry.
I wouldn't be here if I'd realized there was a problem. Usually it's
no, come on.
It was amazing.
Of course not.
And for me, it's a very great honor to have you here, because your papers and your work is so huge and so important. Thank you very much.
There's still so much to do. I think as soon as you
that's the nice thing of biology.
So much to do. It's just so funny and very hard. And every enzyme family you look at is slightly different. They're really complicated. So and it's of course it's with all the genome things. People tend to move a level up and they don't worry about the detail. They just sort of, and it's frustrating really sometimes.
It's true.
Okay, so, thank you, Shannon.
Thank you, Shannon.
Thank you. I will leave because I am screwing up everything. Okay.
No, you are not. Thank you.
All right. Thank you very much.
Have a nice day. Thank you.
Be in contact with you for any further questions and proposals and whatever. Thank you, Shannon. So we are going to
collaborate with people.
We know about that. That's wonderful. Bye, Shannon. Thank you very much.